Hello students, welcome to biology class. This video is in continuity with the previous video of chapter reproductive system. The objectives that we are covering in this video are the male gamete, that is sperm, structure of the sperm, then the process of formation of sperm, that is spermatogenesis, hormonal control of spermatogenesis, and diseases and disorders which are related to male reproductive system. So, the first topic is male gamete or sperm. This is the generalized structure of the male gamete, that is sperm, which contains a head part, the middle piece and the tail portion. Structure of male gamete, sperm. Sperms are highly specialized cells. They are produced in the seminiferous tubules of the testes by the process called spermatogenesis. Each sperm is a microscopic, elongated, flagellated and motile cell. Human sperm consists of the following four parts. Head region. It is slightly oval and contains large nucleus which has a highly condensed DNA matter. The nucleus contains haploid number of chromosomes. Over the anterior half of the nucleus is present a cap-like structure called acrosome, which is actually a modified Golgi body. The acrosome contains enzymes necessary for fertilization. Next is neck. Neck. It has a pair of centriole. One centriole is known as proximal centriole and another is known as distal centriole, present at right angle to each other. From one of the centriole emerges the fine fibrils or microtubules that run through the middle piece and form the axial filament of the tail piece or the flagellum. The middle piece. Middle piece contains mitochondria in a spiral form and microtubules of the centriole. A ring centriole is present at the junction between the middle piece and the tail piece, but the function of the ring centriole is not clearly understood. The last is the tail. It is a large whip-like structure and it consists a specific arrangement of microtubules, that is 9 plus 2, which is typical of cilia and flagella. It gives motility to the sperm. Moving on to the next topic, functions of various parts of a sperm. Sperm consists of head part, neck part, middle piece and tail piece that we have discussed. Structures present, they perform a specific functions. Now, a large nucleus present in the head part contains condensed DNA and has a haploid number of chromosomes. Acrosome forms a cap over the nucleus. It contains hydrolytic enzymes that enable the sperm to penetrate into the cytoplasm of the egg during fertilization. Now, the next is neck, which contains two kinds of centriole. One is proximal centriole and another is distal centriole. Now, proximal centriole plays an important role during the first cleavage division of the zygote, and the distal centriole forms the axial filament that forms the tail piece of the sperm and is attached to the distal centriole. Now, the next is the middle piece, which is having a spiral mitochondria. Now, it contains respiratory enzymes which supply energy for the movement of the sperm. Means it gives motility to the sperm. The last is the tail piece, which is having an axial filament surrounded by cytoplasm and enclosed by a tail sheath. Now, it is a modified flagellum that propels the spermatozoa in liquid medium and in female reproductive tract. Moving on to the next topic, gametogenesis. Gametogenesis is the process of formation of gametes that occur in male and female gonads, that is, testes in male and ovaries in female. In both the cases, the basic sequence of events remains the same. The cells of germinal epithelium undergoes three essential stages. First one, multiplication phase. Second one, growth phase. The third one, maturation phase or meiotic phase to give rise to haploid gametes. Now, spermatogenesis. Gametogenesis for the formation of sperms is known as spermatogenesis, and gametogenesis for the formation of female gametes, ova or eggs is known as oogenesis. Each tubule contains two types of cells, germinal epithelium cells and Sertoli cells. The germinal epithelium cells, which are also called primary germ cells, lines the tubules internally. These cells are diploid in nature and undergoes a number of stages to give rise to haploid spermatids. And the Sertoli cells, the second type of cells, are large elongated supporting cells present in between the strands of developing cells. They project into the lumen and provide nourishment to the developing sperms. The spermatids get embedded in the Sertoli cells and get transformed into sperms. The sperms, however, are immature, hence stored in epididymis where they turn into mature sperms. Now, here we will be discussing the important stages of spermatogenesis that will lead to the formation of sperms. There are mainly four important stages of spermatogenesis. First one is multiplication phase, second is growth phase, third is maturation phase or meiotic phase that includes meiosis 1 and meiosis 2. And the last is spermiogenesis. Starting with the first phase of spermatogenesis, that is multiplication phase. Multiplication phase, the germinal epithelium cells or primary germ cells of seminiferous tubules, also called the spermatogonal cells. These cells are small cells of about 12 micrometer in diameter. At maturity, they undergo repeated mitotic division to form a number of spermatogonia. Each spermatogonia is diploid and contains 46 chromosomes. In this picture, we can see the complete process of spermatogenesis. In multiplication phase, spermatogonia undergo mitotic division, result in the formation of primary spermatocyte. Primary spermatocyte undergo meiosis 1, which is an reductional type of cell division, result in the formation of secondary spermatocyte. Secondary spermatocyte undergo meiosis 2, which is an equational cell division, result in the formation of 4 haploid cells. The spermatids from the secondary spermatocyte have half number of chromosome as that of the parent cell. Spermatids undergo spermiogenesis to form haploid spermatozoa or sperms. The second phase of spermatogenesis or the growth phase. Now, some of the spermatogonia increase in size by accumulating nourishing material from the germinal cells. Now, they become almost double in volume and are called the primary spermatocytes. The growth phase is a preparatory phase for the initiation of meiosis. Now, primary spermatocyte is large and diploid and contains two sets of chromosomes, that is 46 chromosomes. Now the third phase of spermatogenesis is the maturation or meiotic phase, in which each primary spermatocyte which is diploid in nature undergoes the first meiotic division to give rise to haploid secondary spermatocyte. This in turn undergo a quick second meiotic division, that is an equational division, to produce a total of 4 equal haploid spermatids. Now each spermatid has 23 chromosomes. Now the spermatids are unspecialized cells and do not undergo any further division. All the 4 spermatids, though have separate nuclear material, still remains connected by the cytoplasm. They separate out only during the process of spermiogenesis. Now entering into the fourth stage, that is spermiogenesis, in which the spermatids differentiate into sperm. It is the transformation of an unspecialized spermatid into a spermatozoa or sperm. Spermatids get embedded in the Sertoli cells, which provide support, protection and nutrition. The spermatid get transformed into a sperm by losing a great deal of cytoplasm, condensation of the nucleus into the head, and by the formation of a flagellated tail. So these are the changes that are taking place in the spermid to differentiate into a sperm. Now here is the process of spermiogenesis, which is differentiating the spermatid nucleus into the sperm. Now during development, the flagellated tail of the sperm projects into the lumen, while the head is embedded in the Sertoli cells for the nourishment. Mature spermatozoa or the sperm finally gets detached and is released in the lumen of the seminiferous tubule by the process called spermiation. From one diploid primary spermatocytes, 4 haploid sperms are formed. In this image, we notice the transformation of primary spermatocyte into a spermatid, which later transform into sperm. Now, temperature is very crucial factor for the development of sperms. For any reason, if the testes fail to descend from the abdominal cavity, spermatogenesis does not take place and leads to sterility. An adult man produces about 10 raised to the power 12 to 10 raised to the power 13 sperm cells in a day. The sperms gradually move into the epididymis, where they get concentrated, undergoes maturation and are stored. Sertoli cells secrete an androgen binding protein ABP that concentrate testosterone in the seminiferous tubules. Now, moving on to the hormones of male reproductive system. Hypothalamus. Hypothalamus produce gonadotropin releasing hormones, which stimulates interior pituitary to release follicle stimulating hormone FSH that initiates spermatogenesis, and luteinizing hormone LH that stimulates interstitial cells in the testes to produce testosterone. Testosterone further bring secondary sexual characteristics in males and maturation of male reproductive organs, and also it is regulated by negative feedback mechanism. Next we are discussing hormonal control of spermatogenesis. Spermatogenesis start at the age of puberty. Puberty, it is the time period when the sex organs become functional. That is between 13 to 16 years of age in boys. The gonadotropin releasing hormone is a hypothalamic hormone. It stimulates interior pituitary to produce 2 gonadotropins. First one, luteinizing hormone or interstitial cell stimulating hormone ICSH, and second one is follicle stimulating hormone. LH or ICSH stimulates the Leydig cells or interstitial cells to secrete androgen like testosterone that is essential for spermatogenesis. FSH stimulates the Sertoli cells to facilitate sperm development or spermiogenesis. FSH also stimulates Sertoli cells to produce a peptide hormone inhibin that specifically inhibits FSH secretion. Rising level of testosterone suppresses the release of gonadotropin releasing hormones from hypothalamus. Under the control of FSH and testosterone, Sertoli cells secrete androgen binding protein that concentrate testosterone in the seminiferous tubules. Both testosterone and inhibin exert negative feedback inhibition on the secretion of LH and FSH respectively. This image shows the hormonal control of spermatogenesis. Hypothalamus produces gonadotropin releasing hormone, which stimulates anterior pituitary to produce two tropic hormones. First one LH and second one FSH. LH or luteinizing hormone stimulates Leydig cells to produce hormone testosterone, which is essential for the process of spermatogenesis. FSH or follicle stimulating hormone stimulates Sertoli cells. Sertoli cells help in the development of sperm and in the process of spermiogenesis, and Sertoli cells also produces inhibin which gives negative feedback to anterior pituitary. Both testosterone and inhibin gives negative feedback to the anterior pituitary for the secretion of FSH and LH. High level of testosterone also suppresses hypothalamus for the release of gonadotropin hormone. Now, in the last, we are discussing diseases and disorders of male reproductive system. The first one is benign prostatic hypertrophy, that is BPH, which is a non-malignant enlargement of the prostate gland common in older men. The second one is epididymitis, that is the inflammation of an epididymis usually starts as an urinary tract infection. The third one is impotency or erectile dysfunction, ED, the disorder in which erection cannot be achieved or maintained. About 50% of males between 40 and 70 years of age have some degree of ED. The next one is prostate cancer. Most common form of cancer in men over 40 years of age. Risks of developing it increases with age. The next one is prostatitis. That is, inflammation of prostate gland may be acute or chronic. The last one is testicular cancer. Malignant growth in one or both testicles, more common in males 15 to 30 years of age, and more aggressive malignancy. So, we have discussed the complete male reproductive system, including the different organs and the formation of sperm and the diseases which are related to male reproductive system. Thank you so much.